welcome back to the discovery track. We're about to resume here. So before we start, let's see. So yes, thank you for joining us on this continuated discovery track. It's my pleasure to introduce our second speaker for this session, Ylva Terelius, who is CEO at ADMET AB in Sweden. Ylva will share with us her experience with the use of ADMET Predictor in early discovery, drug metabolism, and pharmacokinetics. So without further ado, Ylva, the floor is yours, and thank you very much for presenting in the session today.
Thank you, and I have to thank the organizers for inviting me to talk about our results. And actually, this presentation is earlier than the previous presentation because this is really early in projects and often in small startups. They don't have money enough to screen a lot of compounds. So the question was if we could actually predict the solubility and metabolic stability and permeability from virtual compounds. So we presented a paper last year, Anna-Karin Solenius, Stanbeck, and I, and it's published in Drug Metabolism and Disposition, and you can find more details in there. So why do we want to predict anything in early drug discovery? Well, of course, predictions of basic compound properties from virtual structures can help prioritize synthesis and testing and thus save cost and time for projects. But to filter out compounds, the predictions need to be very reliable. You don't want to throw away the baby with the bathwater, so to speak. So you can't rely completely on predictions. So a few compounds with poor properties should be made and tested along with the best compounds to just verify that the predictions are correct. And then you have to, of course, note that there's always a compromise to be made between physical properties, ADMET properties, potency, and toxicity. So sometimes you will have to choose something that is not in ADMET that is really efficient and potent in vivo, for instance, in an animal model or so. So I want to present the characteristics of the data set we used. It was compounds from Medivir company, and we were not allowed to publish the structures, but we were allowed to use the data, the predictions, etc. So it was 4,794 compounds, which had measured HLM Clint values. And they were mostly protease and polymerase inhibitors, which are not really representative of the whole drug chemical space. So it's a little bit off. And then we wondered if it would work with the global models from ADMET Predictor. The compounds were fairly pure. Most of them were over 95% pure, and none were below 80%. And we removed all unstable or insoluble compounds. They were not included in this work. So the 4,794 compounds were the ones remaining after removing the unstable and insoluble compounds. And they were then a mix of citrate ions, weak bases and weak acids, a few neutrals, and a couple of mixed pK. And the data we had measured with fairly high quality, I would say, was HLM Clint values, and they were the total Clint values. We had log D from some compounds, not all of them, but it was from the same compound set. So it's not other compounds, it's from the 4,794 compounds. We had 1198 that were measured in log D. And 2,778 compounds had measured solubility values. And 2,586 compounds had measured CACO2 values. We worked then with ADMET Predictor version 9.5, and we used the SIP HLM Clint model, and that was unbound Clint. So we actually converted that to total Clint using the Simulations Plus fraction unbound in microsomes. Because if we tried first to correct our total to unbound, but then we had to use the predicted FU values in that direction, and we felt that was introducing some more complications and faults into our data set, since we didn't know the quality of that model. And for log D, we used the Simulations S plus log D model, and solubility, we used the Simulations Plus water solubility at the pH 7.4. And for CACO, at that time at least, there wasn't any CACO2 model in ADMET Predictor. And now at least you can predict CACOs in GastroPlus and Membrane Plus and so on, and maybe in ADMET Predictor also. I haven't looked at the latest version. But we tried to compare that with the S plus PEF instead, because that's the efficient jejunal permeability. And we thought that that could possibly work as an early screening, simulation screening for very early projects. And the reason we are talking about really early projects is that at the moment, I am mostly working as a consultant for extremely small companies or startups, and they don't have any resources to synthesize and test a lot of compounds. So if we can filter out compounds and prioritize at the very early stage, their money may be sufficient to do some testing. So also in the characteristics section, we have the molecular weight and log D distribution for this set of compounds. And all compounds, of course, have a molecular weight. So that's in the graph to the left. And you can see the median and average values there as well. And the measured log D values are to the right, and they are for the 1198 compounds that had measured log D values, and the median and average are listed there as well. So just a little bit about the assays. You can read this later, since you will get copies of these slides, and you can also read the publication. But the log D is kind of standard. It's just a 15 micromolar final from a 10 millimolar in DMS stock. And it was vortexed with octanol and phosphate buffer and allowed to settle and centrifuged. The solubility you can measure in several ways. And in this test, then we used only kinetic data. And they were 100-fold dilutions from a 10 millimolar stock. So they were 100 micromolar in the final sample. And since they were only 100 micromolar to start with, we couldn't get higher solubilities than 100. So we wrote everything that was completely soluble. We just wrote above 100. Yeah. And for the permeability, the measured permeability, we used CACO2 cells from ATCC at passage 36 in 96 well plates and cultured for 21 days. And the incubations were for 2 hours. And we had 1% bovine serum albumin in the basolateral buffer. And you can run that assay in different ways as well. So it's important to know the conditions, but also there are cell-to-cell batch variations. So in this case, we tried to use same, and also we used, for all the assays, we used reference compounds to check for both reproducibility and the quality. And models for some of the compounds, we did also a plus-minus PGP inhibitor experiment, or ABBA experiment, where we looked at the permeability in both directions. And we actually tried to compare that to the Simulations Plus PGP substrate yes-no predictions. And for the HLM Clint assay, then we used 1 micromolar compound and 0.5 milligram protein per milliliter in 100 millimolar phosphate buffer at 7.4. And we made the whole time curve from 0 to 45 minutes, and then fitted the disappearance to first order elimination equation. But we didn't have unbound fractions measured, as I said, and so we did this conversion of the unbound Clint values from ADMET Predictor to total by correcting for the predicted S plus FU MIC. Okay, so for some assays, we tried to make scatter plots, etc., but we found it was more useful actually to make bins, and we made 4 bins out for every assay or for every data type. And you can see here that we have numbered them A to D, and here they are listed so that the lowest numbers are in A and the highest numbers are D, but that means that for solubility, CACBO and PEF, the lowest numbers are the bad ones and the highest are the good ones. But for Clint values, the lowest value is the good one and the highest is the bad one. And the reason for 4 bins is that we wanted to know which ones were good and which ones were bad, and we wanted to separate them. And then in the middle, we thought it would be good to divide that anyway, so that we know if they're closer to good or closer to bad. So that's why there were 4 bins. And then you can see also there how many compounds were measured in each for each parameter, and also you can see the percentage of compounds that appeared in each bin. And for solubility, you can see that the high solubility compounds were overrepresented. So that is not quite a balanced data set. The CACBO2 PAP values are almost perfectly balanced between high and low. And the Clint values, that we have an overrepresentation of the high clearance compounds, but it's not as bad as the solubility. Okay, besides actually comparing directly with the Simulations Plus global models that are there for, originally when you license the program, we also used the ADMET Predictor modular module, which you have to license separately, to build in-house models. And we used the logarithmic build models for solubility, HLM Clint and CACBO2 PAP. The training sets used were approximately 75 to 80% of the available measured data, with the remaining compounds then used as the test set. And the test sets for the local model were chosen by the modular module based on cohonent mapping. And once we had the test sets and we had the models, we of course ran the model with the test set, but we also ran the test set with the global ADMET Predictor models for comparison, the same compounds. And each model was rebuilt at least 4 times, and the deviations were around 10% or less. So the results I'm going to show is from one of the representative models built. So this is a global model with all the measured values for log D. And the R2 value, as you see, is pretty good, I think. It's 0.79. So, I mean, you can rely on that model, I think. So it's a good model. But we also did the in-house model. And with the test set to the right here, B, with a global model, it's the same R2 value, and it looks quite similar to the total data set. So I think the test set is well representing the total data set. And to the left, you see the local model we built in the modeler. And you see that even though, I mean, the global model is good, I mean, the in-house model is slightly better. The R2 value goes up to 0.89, which is very good, of course. Then for solubility at pH 7.4, we did also the predictions. And this slide is kind of difficult to read, so I have to introduce it to you. So the different bins for the measured solubility on the x-axis. So the poorly soluble compounds are to the left, and the highly soluble compounds are to the right. And then within each bin for measured solubility, it says how many compounds there were at the top. So there were 582 compounds with solubility below 10, and 1,537 compounds with measured solubility over 90. Then within those, we saw how many percentage were actually predicted below 10, or 10 to 50, or 50 to 90, or above 90. So those are the colored columns in the diagram. So you can see that there's a trend here that, you know, the predictions are pretty good, but it's still poorly predicting the very poor solubility compounds. So it's over predicting the solubility for our may not be representative for your compounds, but still. Yeah, so we chose a training set then of 2,087 compounds. And then global model tested with these 691 compounds that were the remaining compounds after removing the training set. It looks fairly similar to this, but maybe not quite as representative. And you see that the good solubility is overrepresented in more or less all of the bins. The test set in the local model, however, is predicting the low solubility compounds much better, and not quite as good at the high solubility. But if you put the limit instead at the 50 micromolar, so you add the green and the yellow, then it's getting very good also for the high solubility compounds. But it's actually then under predicting the high solubility, but it's pretty good at the low solubility. And that was our main goal, to be able to weed out the really poorly soluble compounds. And that we can clearly do with the local model, and it's better than the global model. So now I will move on, but I will use the same structure in the coming slides. So it's good if you understood the graph here, that these are the number of compounds measured with these properties. And in here, it's the percentage of those that were predicted in the different categories. Okay. So here we have the similar picture then, or slide for the HLM Clint predictions and models. So we have the total data set here with the 4,794 compounds. We have 1,332 in the metabolically stable bin, and 1,900 in the bin, so to speak. And why we put these limits is that the 15 is really good for most studies, and I would say that the clearance is too high, maybe 50 or something, but we didn't want to weed out too many compounds at this early stage, so we put the highest at the 80. And then, as I say, we wanted to know if the in-betweens were closer to good or closer to bad. So there are 4 bins. And here you can see what the colors in the columns mean then. So it's not bad. I mean, there are quite a lot of compounds that are predicted correctly here, but it's not perfect. I mean, if you add the green and the yellow, then of course you can say that it predicts the low Clint compounds fairly well. And if you add the red and the orange, it predicts the high Clint compounds pretty well. And after training, we had 1,199 compounds left in the test set. And it looks very similar in the distribution in the global model between the different bins. For the local model, however, the correct prediction sort of goes up in the low Clint bin, and it goes up also for the poor Clint compounds, the high Clint compounds. So it's better at the extremes. And we really never used the actual Clint numbers for the 2 bins in the middle, because they were kind of not as clear, and it was difficult to put the sort of border in a correct place to actually use them for calculations and scaling to humans or to in vivo or whatever. But at least we had a better model with the in-house built model than with the global model, but the global model isn't bad. Then we did the same for the permeability, and now it becomes a bit more tricky since there wasn't any CACO2 model available in the ADMET Predictor 9.5. So we had to compare with the PEF value in the model, in the ADMET Predictor. And then we had to use 2 different scales for the CACOs and the PEF. So the CACOs we used less than 2, 2 to 5, 5 to 10, and above 10. But for the PEF, we looked at previous historic data, and we considered that the PEF values that were similar to the CACO values were correct if we binned it like less than 1, 1 to 2, 2 to 3, and above 3. So this first one with all the compounds, then we have the measured PAP values from CACOs, less than 2 to 5, 5 to 10, and above 10 here. And you can see how many compounds were in each bin, and it's, as you see, fairly balanced. And then you can see the global model actually predicted approximately 70% of the compounds correct if we use the PEF with this scale to divide this bin in poor and good and so on. And yeah, it looks fairly nice actually. We could possibly use the PEF model in the ADMET Predictor to actually predict at least the poor and the good permeability compounds, but especially the poor compounds. The good are not quite as good. They're a little bit under predicted. And then we built our local models from 2070 compounds, and the test set then remaining was 516 compounds. And the global model predicts approximately like for the whole data set, so it's probably fairly representative of the whole data set. And the local model predicts a little bit better. It's almost similar for detecting the poor permeability compounds, but the good permeability compounds are better predicted with the local model than with the global model, you can see. But then remember that this slide, or the graph up at the upper right, there we have the measured CACO against the predicted CACO, because we built the model for predicted PAP and not PEF. But here we compare the measured CACOs with the predicted PEF which is the same as we did for the global set. Okay. So then for the poor permeability compounds, we also looked then if we could identify if they were PGP substrates or if they were just poorly permeable for other reasons, like for instance if they were too hydrophilic or so. And we had 387 in-house compounds that were tested in the A to B with and without a PGP inhibitor. And a total of 264, that is 68% of these tested, had a plus-minus inhibitor ratio of above 1.5, which we considered as an indication of efflux. We also had 103 compounds tested in the ABBA assays. We looked at 94 compounds, but there 91% of the tested compounds had an ABBA ratio above 2 because we tested mostly the poor permeability compounds. So that data set is really skewed. It's not balanced at all, of course. So we did a confusion table where we plotted yes, no for the predictions and yes, no for the assays. And when we looked at the result and combined those, we got this result. And so that the overall accuracy was 0.75 for if we compared with a PGP inhibitor assay, and 0.86 if we compared with the ABBA above 2 assay. But of course, as I say, the ABBA above 2 assay compared to the predictions could overestimate the precision and accuracy, since almost all of them were yes.
I'm going to ask you to conclude since we're about 2 minutes out.
Okay, sorry. I tested this before and then I was much faster. So, okay. Sorry. We also actually tested this model as at least for reference drugs, and the reference drugs are listed in the supplemental data in the publications. You can see which ones they were. We had 75 reference drugs for HLM Clint and 26 reference drugs for CACO2 values. And you can see here that in the HLM Clint predictions, the local model gave a better R2 than model, even for the reference drugs, which surprised us a little bit because we thought they would be included in the training set for the global model. And this model is very good, the CACO2 model. Yeah. So you have a summary, but the take-home message then is that the data set is kind of odd since it's mostly protease inhibitors. So that is a difference why the local model could be better than the global model. But we can almost always improve models by building local. And the reasons for improved predictions are probably that we have a better representation of the structures that we're interested in, but also that in this case, we actually use the same batch of pooled microsomes, for instance, for all assays, etc. So the assay systems were similar. So I have also actually built local models to just 20 compounds from a chemical series. And that gave a very good model that worked for the next 5-10 compounds. But then you had to rebuild it because then you were outside the chemical space of the model that you built. But that is possible. So I want to just thank the former chemists and the DMPK staff at Medivir for producing the data, and for Medivir for allowing us to use this data set. And thank you for listening. And I'm sorry I was a bit...
That's all right. Thank you so much for this great talk. Unfortunately, we don't have time for questions. No. This concludes this part of the discovery track. We'll be back at 11 a.m. Pacific time for more, with a poster presentation and the presenter panel discussion. Thank you so much, and We'll be back in just a little bit. Have a great rest of the day. And thank you so much for presenting.